whether or not probiotics can help with AHPND, EMS, and white feces. This is a slide that has been shown recently by one of the major Thai company, CP, to show that after EMS, shrimp culture will never be the same again. And we had a nice presentation earlier by Dr. Soto. But some of the key points here I would like to emphasize is the fact that now you have many different strains of Vibrio parahermeticus involved, but also that the genes associated with EMS, HPND, are now being found in strains of other species. It was mentioned EMS in Australia. In Australia, it's not Vibrio parahermeticus, it's another species of Vibrio, which is associated with the EMS outbreak in Australia. Also, we now have reports of milder isolates in Mexico. They showed a very aggressive Vibrio that killed in a few hours. Some of the strains we have in Vietnam or Thailand are not as aggressive. And other are thought to be maybe linked to white feces. So not giving HPND, but something else. Finally, Flegel and other people in Thailand have reported other bacteria, other species, Shewonella and a few others, that could be associated with EMS, meaning not only Vibrio parahermeticus, but other species could work in synergy to lead to the outbreak. Actually, it was mentioned earlier, but I had prepared that to show the main problem bacteria exchange genetic material. They can do it by just incorporating genes from one species to the other, or they can do it by transferring this genetic material, which should be on a plasmid, which is the case of the Vibrio parahermeticus associated with EMS. So that means you have high probability of these genes being exchanged. And this is why it's not only screening for specific Vibrio parahermeticus, but more maybe overall the bacteria that could be associated with Vibrio strains. White feces. This is something that is giving problem in India, in Indonesia, in Thailand for many years. The cause is not so clear. In the past, it was associated with poor quality of feeds, mycotoxin and so on, but also has been associated with Vibrio. If we look in the literature over the last few years, you can find the list of Vibrio species which are associated with white feces in Indonesia, in India, in Thailand, and in China. So that means many of these countries are affected both by EMS, but also white feces, and Vibrio species are found in both cases. Okay, no need to look in detail for this. What it matters is to talk about the mechanism of communication of Vibrio, quorum sensing. This is important because it's the mechanism the Vibrio use to understand whether or not they reach a certain number, abundance of bacteria, and then they become pathogenic, virulent. What you can see here is that the same kind of molecules, signal molecules, are found in a large number of species of Vibrio. And these are all associated with disease and virulence. So, one more time, to control EMS, it's not only about controlling one or two strains of Vibrio parahermeticus, it's controlling much larger communities. So, how do we cope with Vibrio? That means looking at solution both from broodstock, hatchery, nursery, and grow out. It's easier sometimes to just blame the supplier of the PLs or the hatchery blaming the broodstock supplier. Actually, we have to integrate all the steps. At the beginning of AHPND EMS in Thailand, Flegel and colleagues made a nice survey to show that the live food used in broodstock was a big part of the contamination. So at the end, the recommendations were reduce the use of live feed and screen the broodstock. Now in Thailand, they have also microsporidium EHP, so it's an extra step that is to be taken to screen the quality of the broodstock to make sure you don't transfer your pathogen from your broodstock to the hatchery and so on. If we look at the hatchery, there again, it's important to identify all the possible points of entry of the Vibrio. For example, the algal culture, there are some simple treatments, very easy, that can be done to control the Vibrio in the algal cultures. Same with the Artemia, to prevent the Artemia being a source of Vibrio for the PL, or for the myces, and also to control Vibrio biofilm. It has been mentioned also earlier, Vibrio, like most bacteria, create biofilm. Biofilm gives them more strength and the ability to resist against disinfection, antibiotic, and so on. So there are also methods that can be used to control the biofilm and therefore prevent the Vibrio from recolonizing other facilities. During the culture, now the use of probiotics to control Vibrio during the larval rearing is, I think, very well accepted. There's a lot of publication, peer-reviewed, to document how you can control the Vibrio in Zoea, myces, or PL, which allows you to make sure that when you transfer your PL from the hatchery to the nursery, you actually reduce the number of Vibrio and make sure you don't contaminate your nursery or your farm. Another step at the bottom part is to work on treatment. So you can work with some plant-based product to clean your PL, or you can work with some treatment with biocide that can also allow you to re-clean the animals before they are transferred. Finally, it's important again to look at the quality of the PL before they are transferred to the farm. This is something in Asia many people stopped when they started working with Vaname. They looked at Vaname as very easy shrimp, no need to check anything. We just get fast growing animals and we just produce. And they stopped looking at all this quality. With EMS, it came back. So now in Thailand, for example, the Department of Fisheries provides free screening. So the people can have their animal checked by recognized labs. Many also laboratories provide this service. That means you are sure your animal is checked. The new one now is, of course, EHP, which is a major problem now. But then also the standard analysis to make sure you have quality animals. Now, when we look in nursery, when EMS came, the interest in Asia was to study the concept of nursery, and basically Mexico. Unfortunately, nursery by itself was not a solution, as Mexico got hit by EMS. But what we have shown through working with companies in Mexico is that extra probiotic treatments to the normal protocol can give you an improvement on survival, but also on the growth of the animal. So if you select bacteria that can compete with the Vibrio, and you apply these in the right way in the nursery, you can really have an improvement on the growth and survival of the animal. Now, this is present data from Dr. Put from the Department of Fisheries in Thailand. He presented it a few weeks ago in Thailand. It's very interesting because he looked at all samples from broodstock all the way to farm, and look for the plasmid of EMS and the toxin genes. And what they saw is that the farm water, the farm sediment, and the PL were the main source of contamination. So the treatment of the PL and juvenile, very important. That means screening and emphasis on quality in the hatchery. But then you need to look at the farm sediment. So when they observe that, the approach that can be taken is how can we control the sediment to avoid the Vibrio being dominant. First point, all Vibrio like chitin, the mold of the shrimp. So to remove this mold or to avoid the accumulation inside the pond is the first thing, to make sure you don't have support for the Vibrio. Second, Vibrio are very fast growers and they have the ability to stay in a state of not dormancy but slower activity. When there is nutrient, after algal crash or something else, you suddenly have nutrients coming and the Vibrio will be the first one to grow fast and colonize. So it's very important to avoid accumulation of the mold, but also keep the quality of the sediment to avoid having nutrients for the Vibrio. So you can see two ways. This is some of the new intensive system in Thailand. As you can see, it's a lot of piping, water exchange, flushing out continuously the waste, redesigning of the pond to concentrate the waste and flushing out non-stop. This is very expensive. It requires a lot of investment. If you are part of a big company, you can do that and it works. But for some, the medial player is much more challenging. And there are also issues that you produce a lot of waste that you release in the environment. So it's another point to consider. The other way is basically closer to what maybe is done in Latin America. It's looking at ways to control the quality of the soil with probiotics, but also, when required, do some flush. This is coming from super intensive in Indonesia. And as you can see, they have a central drain, they flush out, and the workers collect the mold and they spread the mold here. So they remove the mold and they observe if there are signs of disease, of mortality. So it gives them an idea if there is a beginning of an outbreak, and then they can do some treatment or modify the management. Now, how can probiotics help to control the environment? Probiotics can be selected for the production of enzymes. There are many enzymes. These are some of the enzymes that we focus on, and we work with the protease, amylase, cellulase, and so on, because they can help to degrade algal material, but also uneaten feed ingredient or dead animals and the feces. So it's a tool to allow you to degrade the waste. It's also important to explain how this is done. So when we register product, we need to go to an external lab. This is the method they use, the substrate used for the test, and it means we can explain how we can claim, why we claim we have these enzymes and how they work and the activity. This can be done with all registered, accredited laboratories. Once we select probiotics to degrade the waste, the consequence is that we want to have less waste in the pond. If we have less waste in the pond, as was mentioned also earlier, less nutrient for the Vibrio to grow, lower probability of anaerobic conditions, less stress for the shrimp, and also lower production of ammonia sulfide and so on. So less Vibrio, less chance of Vibrio growing, and also less stress for the shrimp. These are data from Brazil to show that if you apply probiotics in the water and in the feed, you can remove the Vibrio from the sediment. In this case, this is something we did in 2007. So we've been working on it now for over 12 years. We have data worldwide. And we can show that the bacillus, in this case, can be recovered from the pond bottom, because this is where everything happens, while the Vibrio can be displaced. It was important because when people looked at that first, they were focusing on the water, and they could not see any trend. And they said, your product don't work. I check the water, no difference. But bacteria go where there is substrate, where there is nutrients. It's in the pond bottom. So it has to be effective in the sediment. Now, when we look at white feces or EMS, it's important to include the shrimp gut treatment. So in this work here in Thailand, they looked at Vibrio havei and how it could be recovered from different parts of the gut and be associated with the disease. EMS, whether or not it colonizes the stomach or it just accumulates there, it's another area where we need to find a way to control the Vibrio from the gut of the shrimp. How do people do that? First, you can select probiotics that can kill the Vibrio. So in this case, this is a strain of Bacillus. These are Vibrio parahemolyticus from a culture collection. So you can buy them and test yourself. And it shows you inhibition. This is Vibrio havei, and you can do a different method to show that your Bacillus kill directly the Vibrio. The second point, which maybe is more important, is how the probiotics, when they are put in the feed, can displace the Vibrio from the gut. It's work that we did also almost 8-9 years ago. And it shows you the Vibrio in the gut of the shrimp, yellow or green colonies, and then you start to put the probiotics. No more, you stop, it comes back. It was done in Australia. The message is, if you can occupy the gut with the bacillus, you will have less chance of the Vibrio being present. So you can reduce the Vibrio by just occupying. This was done with traditional methods. Now we work with more high-tech techniques. This is work done by a team in Brazil, a PhD student from FURC in Brazil, colleague of Doriano, and she worked on seeing how probiotics can reduce Vibrio in the gut of shrimp, whether you work with biofloc or traditional. And what it shows you is that if in biofloc you put the bacillus in the water, you recover them from the gut of shrimp. It's normal. In clear water, you recover them also. And at the same time, if you look at the Vibrio, if you put the probiotics, you reduce the number of Vibrio in the gut. If you go clear water, you reduce. So the message is, it's possible, whether you work in biofloc or in clear water, to reduce the number of Vibrio from the gut of the shrimp. So we can do it in the sediment, but we can also do it inside the shrimp. You saw a graph of challenge test earlier. Is another one. This one is done in Vietnam. In this case, we provide the probiotics in the feed, and then we challenge the animal. This is the mortality of the shrimp with the EMS. In this case, for the challenge test, it should target 50% mortality. This is the goal. So we adapt the dosage of Vibrio to reach 50% mortality. Then the next step here, you do nothing, no challenge. no mortality. In green, it's antibiotic in the feed. And you can see you can improve by reducing mortality. In here, this is the probiotics in the feed. And you can have even a further improvement. So it's logical. If you remove the Vibrio from the gut, you have less chance of this Vibrio reaching a large number enough to create the disease. Other benefit that you can see from probiotics in the feed is they will reduce the FCR. What it means is, okay, lower feed cost, which is good, but also less waste in the pond bottom, since the food is taken up instead of being wasted. So one more time, less chance of Vibrio growing. And second, the bacillus will stay in the shrimp feces. So these feces are important because the shrimp eat their own feces. So if you colonize the feces, the Vibrio, which are fast grower, will have less chance of dominating the feces. You have less waste, and again, less chance of Vibrio. So it's additional ways in which we can control. So if I reach some conclusion for this, the reason probiotics can help for EMS or white feces, the first one is controlling the waste, so less chance of Vibrio growing. They can kill directly the pathogen, but they can also improve the feed utilization and therefore less waste in the pond bottom. So one more time, reducing the growth of Vibrio. And what I showed you, they can occupy the environment, whether it is the pond sediment or the shrimp gut. So these are all angles that allow us to reduce the Vibrio. It's not to remove them, it's just to maintain the Vibrio at an acceptable level. But we have to insist, it's not the magical bullet. Probiotics cannot solve everything. It's just one tool for the farmer. And that means, this is why I emphasized at the beginning, it has to go from the broodstock, hatchery, nursery, and so on. And prevention of transfer from each step. So it's only through this holistic approach that disease like EMS or white feces can be handled, and that the probiotics can be part of it. Thank you very much. Muchas gracias.
Thank you, Olivier, for your presentation. This is for you. Since all of you had a really good time, ya que él tuvo un muy buen tiempo, vamos a tener un par de preguntas si alguien quiere consultarle algo a Olivier. O quedaron bien claritos o bien confundidos. Gracias, Olivier. Gracias. Como última presentación de la jornada de la mañana, vamos a seguir con una charla referente al tema de genética. Vamos a invitar al doctor Richard Towner, egresado de la Universidad de Colorado en Estados Unidos con una maestría, doctorado en la Universidad de Madison en Wisconsin en Ciencias y Genética, miembro de la Sociedad Americana para la Salud Animal, también miembro de la Sociedad Mundial de Acuicultura, miembro de la Sociedad Americana sobre Avances de Ciencia. Si continúo con el currículum, yo creo que nos van a dar la toda la tarde. De momento
Shrimp performance is based upon many factors. It includes genetics, health, nutrition, and management. And to have optimum performance, all of these different aspects need to be at an optimum level. If any one of them has a weak link, then the shrimp performance will suffer. When we look at shrimp genetics, we not only talk about disease resistance, there's growth, reproductive performance, fecundity, fertility, hatchability, etc. So genetics is a composite of many traits that influences the animal performance. But today I'm going to concentrate on the genetics of health and disease resistance. Why is it necessary to have genetics or breeding for disease resistance? It may not be possible to eradicate all the diseases from the environment. When diseases are present, there needs to be some component of genetic resistance in order to improve the survival and improve the shrimp performance. Biosecurity programs can be used to reduce the concentration of the pathogens and reduce the influence of the pathogens on shrimp performance, shrimp mortality. But biosecurity programs are not 100% effective all the time. In many cases, there are vaccines available that will protect animals from diseases, pathogens. But vaccines are not 100% effective all the time. And also it has been shown that animals that do have genetic disease resistance do respond better to vaccines. So genetic resistant animals do have a better response to vaccines and have better performance. I want to show some examples where selection for genetic resistance has worked. This is a case of selecting for IHN in rainbow trout. The selection program was started in 1995. Each of the vertical bars represent the mortality from individual families. You can see that some families at the beginning of the selection program had nearly 100% mortality, and the very best families still had 35 to 40% of the families. So there's a wide range of mortality in the different families. The selection program was started in 1995, and in 2005, the results were dramatically different. There is still a few families that had high mortality from the IHN, but there are many families that had zero mortality or close to zero mortality. Then to compare the two different generations, the red bars show the mortality from the 1995 generation. The blue bars show the mortality from the 2005 generation. There's a large improvement, and this is an example of how genetic selection works. In the majority of cases, genetic selection does not happen immediately. It happens over time, and slow steps are made each generation. In another example, this is from trout also. There is a disease called cold water disease that is caused by a bacteria. In 2002, the mortality was very high in the different families that were tested. The red bars represent the mortality from the 2002 generation. Then the blue bars show the mortality in the challenge test for the 2008 generation. So there's a marked improvement in mortality, a marked reduction in mortality in the 3 generations of selection. The company I consult with has a variety of challenge programs to measure disease resistance. One of those is challenging for TSV resistance. In 2010, the TSV resistance was nearly 80%, which is very good, but we continued the selection, and in 2 generations we were able to increase the survival to about 90%. But since 2012, we have not improved the resistance or the survival to TSV in the challenge test. Some of it is because we're not able to identify some of the susceptible alleles with 100% accuracy. And the other part of it is when we reach 90% survival, we wanted to intensify the challenge tests. So instead of having a 2-week challenge trial, the trial was extended to 3 weeks, and also a higher concentration of the pathogen was fed to the shrimp to make it a more severe challenge, try to create more family differences. In 2010, started selecting for resistance to IMNV. The first generation, the survival was about 15%. 2 years later, in 2012, the survival was nearly 40%. So with quantitative selection using family methods, family data, we were able to increase the survival in the challenge test from 15% to 40% in 2 generations. The reason we stopped testing for IMNV resistance was we wanted to focus on other diseases that were more widespread. In 2011, we started looking at our lines and ways to improve resistance to white spot. At that time, the lines were very susceptible, and if we used the standard challenge test, we'd have essentially 100% mortality at the termination. So in order to create family differences, we terminated the challenge test at 50% mortality. In 2012, it took a little over 2 days to reach 50% mortality. In this past year, this current year, we're now up to 5 days to reach 50% mortality. So we've been able to increase the resistance to TSV. It has been a slow process, but the process we're using is working. EMS is another important pathogen, severe pathogen in many parts of the country. In 2013, the University of Arizona developed a protocol for a challenge test for EMS. We did some testing in 2013, and in 2014, we were able to start the family selection for EMS resistance. Over the last couple years, the number of days to reach 50% mortality has increased slightly, but what we're going through right now is the frequency of the resistant genes, and this particular population is low. So we're working on increasing the frequency of the resistant genes, and once they reach a certain level, we'll be able to make larger improvements. When setting up a program for disease resistance, there are many different considerations that need to be many different factors that need to be considered. One of the factors is whether to have a SPF population or have a non-SPF population, or commonly called SPR. The different things to consider in deciding the difference or whether to use a SPF or a non-SPF population is the potential for the introduction of diseases. When a person tests the animals in a pond and brings them back to the hatchery for reproduction, there's a potential to introduce new diseases. And also, when you bring animals from the pond back to maturation, there may be other diseases that you do not know about that may influence the ability to reproduce a new generation, or the new diseases may cause problems in future generations. It is shown that many of the pathogens are vertically transmitted either through the eggs or attached to the eggs. So if a person is dealing with a group of animals that is non-SPF, there may be a possibility of vertical transmission and infection of the offspring generation. Typically, many companies use SPF populations in the breeding programs because they need to ship internationally. Many countries require a health document stating you're free of certain diseases, and the only way to be free of the diseases is to have an SPF population. Another consideration in starting a selection program is whether you use wild stock or a domesticated stock. Using wild stock, you can bring them in to a hatchery, quarantine facility, test them for the pathogens, then start using them or developing them for a breeding program. But there is a domestication process that need to go through. There are many wild animals that will not reproduce in captivity, and they are also another group of animals that do not survive well in captivity, so the mortality is very high. When wild stock is used to start a breeding program, the attrition rate is very high, so companies need to start with 3 or 5-fold more wild stock animals than they do domestic stock. And the advantage to the domestic stock is they've also already gone through this process of acquiring or adapting to living in culture conditions, so the selection programs are more effective initially. Typically, the resistance to a specific pathogen is about 10% genetic and 90% non-genetic. So this means that if a person goes out to a pond and collects 10 animals that are alive at the end of the culture period, only one of those animals will be alive because it has genetic resistance. The other 9 of the 10 animals will be alive because they were not exposed to the pathogen. They had excellent feed, excellent water conditions, etc., that allowed them to survive even though they are susceptible. So with only 10% of the differences between animals being genetic, one really needs to look at the accuracy and how to increase the accuracy of selection in order to make more genetic progress. One way to increase the accuracy is to look at families and use families as the basis for selection rather than individuals. This slide shows the increase in accuracy using a different number of individuals per family. With mass selection or a single individual, the accuracy of selection is 0.1. If you test 50 animals per family, the accuracy of selection is 0.4. Testing 100 individuals per family, the accuracy is about 0.45. So it is possible to identify those families that have genetic resistance, and by selecting the families with genetic resistance, a company is able to make more progress than if they use individuals. Another factor involved in genetic improvement is what portion of the families will be selected. If 100% of the families are selected, you're not going to make any genetic progress. But as a smaller and smaller percent of families are selected, the genetic progress will increase. If 50% of the families are selected, the genetic improvement for one pathogen will be approximately 2% per generation. If 20% of the families are selected, the genetic improvement will increase to 4% per generation. So the first thought is to select a few or a lower percentage of the families for reproduction. But the problem of it is, in order to control inbreeding, you need to have a minimum number of families producing each new generation. A minimum of 30 to 40 families are needed to produce the new generation. So, if 50% of the families are being selected, then that means that 60 families need to be produced and tested each generation. If the selection intensity is 20%, then 150 families need to be produced in order to have 30 selected families. So in that case, going from 2% improvement per generation to 4% improvement per generation, a company needs to produce 150 families for testing instead of 60. So there's a 2.5-fold increase in the number of families produced in order to gain the 2% improvement in resistance. And there is no hard and fast rules what the selection intensity needs to be. It varies for each situation, and each situation is different. But the important thing to remember is that family selection is more effective than mass selection or individual selection. What I did to illustrate this is I did a computer simulation using the same parameters for a mass selection program and for a family selection program. And I ran the simulation for 10 generations. After 10 generations of mass selection, the livability improved about 3%. By using family selection, testing the same number of individuals, the improvement was 10%, or 3 times as great as doing the mass selection. Another question in selecting for disease resistance is whether to use a pond challenge or a laboratory challenge. The advantage of a laboratory challenge is you can expose the animals to one pathogen at a time, and you can select for that resistance to that pathogen specifically. Ponds, there are typically multiple pathogens present, and between different batches, the disease load into ponds varies. So one time you may have very high mortality, so the selection for survival is very good. The next time the mortality may be low, so the selection intensity is not as effective. Also, one time there may be a high concentration of Vibrio bacteria, so you're selecting for EMS resistant. Next time you may be selecting for white spot resistance. So with ponds, the challenges are not consistent from batch to batch, but they can give you a broader type of disease resistance. The conclusion is the laboratory challenges is the preferred method to improve disease resistance if you want to improve the resistance to a specific pathogen. If you're looking more for general survival, then a pond trial is better. But there's nothing to say that challenge trials, laboratory challenge trials are mutually exclusive from pond trials. They can be used in combination. Even though a person is selecting for disease resistance, each time you pick an animal to reproduce, you're selecting for a multiple set of traits. And there are relationships between those traits. So if you're concentrating on improving white spot resistance, you may actually be lowering resistance to EMS. Also, if you're selecting for white spot resistance, you may be lowering the growth rate. So there are always trade-offs, and with any selection program, you need to balance what you're doing and consider the other factors. And in most cases, you need to do a multiple trait selection program to counteract some of the negative associations between traits. To evaluate the different alternatives for selection programs, I made up this table. The columns are the sources of the broodstock. You can start out with Wild's broodstock, SPF broodstock, or non-SPF. Considered were disease resistance. What is the result or the risk of having negative tests? What is the risk of being exposed to future unknown diseases? Can inbreeding be controlled so the selection program is long-term? What are the relative weights of disease resistance, improvement in resistance? What is the cost of the facilities and labor to have a selection program? Are there any risks from group spawning? Can you do multiple trait selection? And also, what is the cost of family identification? The red squares indicate a characteristic that is undesirable. The green squares are desirable. For disease cleanup, when you're dealing with the SPF population, it is done one time or a couple times then it's just a monitoring to maintain the disease status. With pond selection, you need to do it every time animals are brought into maturation, so you are not bringing in a whole host of diseases and causing future problems. With all the testing involved, the risk of negative tests is higher using pond selection than SPF populations. SPF populations have a low risk of contamination of other pathogens where there's a possibility with pond selection. In many cases, I'm not as concerned about the diseases that I'm aware of and testing for. What really concerns me are those diseases that are unknown that may be a problem in the future. The only way to control inbreeding is with families. And you have to use families because what happens in a selection program, at some point in time, there's going to be a few families produced that are superior to the rest of the population. And if you do not control inbreeding and just take a group of animals without knowing the pedigree, you're going to be selecting a high percentage of animals from 2 or 3 families. Then the next generation, the inbreeding increases tremendously. So inbreeding has to be controlled. Disease resistance is generally improved faster with an SPF program than a pond selection type program. This is because with SPF programs and challenge tests, laboratory challenge tests, you can concentrate on specific pathogens and improve the resistance to those specific pathogens. In pond situations, the exposure to the pathogens is variable, and because of the variable exposure, the improvement tends to be a little bit slower. The biggest disadvantage to SPF family type programs is the cost. They are more expensive to set up and run than pond selection programs. When animals from an SPF program are used in group settings for group spawning, the exposure and the risk of spreading disease is a lot lower than with pond selection. In both pond and SPF selection programs, there are many possibilities for multiple trait selection. With the traditional SPF program, the cost of identifying the families is very low because of the use of the elastomers. Using elastomers, it costs just a few cents per animal to identify the families. Where in pond selection, you generally have to go to microsatellites or SNPs, and there the cost becomes in the neighborhood of $10 per animal. So there's a trade-off. To review, in designing a selection program, you must define your objective. Do you want to improve general livability or specific disease resistance? And if you only want to improve specific disease resistance, you're talking about laboratory challenge test. If you want to improve general livability, ponds trials are necessary in some form. And like I said earlier, pond trials and laboratory trials can be used together in a selection program to reach the goal that you have. The next area to consider in designing selection programs is the rate of improvement. What is the percentage of families that want to be selected, need to be selected? How am I going to increase the accuracy of selection to increase the rate of progress? And also, what is the limitation in my facilities on the number of animals that can be evaluated? And the third objective to consider is to control inbreeding. Inbreeding must be controlled if you want to have a selection program that is going to be effective for a number of generations. Inbreeding can be controlled if there are 30 selected families per generation. It is better to have 40 or 50. This slide shows some of the genetic differences that have been created in different strains as a result of genetic selection. Strain A has a high resistance for EMS. It has a high survival when challenged for EMS. Strain C has some resistance to EMS. Strain B has good white spot resistance. Then, because of the selection program or the availability of resistant alleles, strain D has very poor survival for white spot and EMS. So, depending upon the way a selection program is designed and the way it is executed, genetic resistance can be created. So far, I've just talked about using traditional methods to improve disease resistance. But in the last few years, we've heard many things about the use of genomics and molecular genetics to improve traits. Recently, the shrimp genome has been sequenced, so the ability of people working with shrimp breeding programs now have a new tool to use to improve disease resistance. So far, there are genetic markers identified for TSV in shrimp, and also there's a marker identified for white spot resistance in monodon. Currently, there are tests ongoing to see if the same gene confers or produces white spot resistance in Van Emmeline. To give you an example of the changes that can occur by the use of molecular genetics, I want to pull an example from the Atlantic salmon. The Atlantic salmon genetics work is more advanced than it has been in shrimp. A few years ago, they identified a resistant gene for a disease called IPN, or infectious pancreas necrosis. A few years ago, the mortality to IPN was 40%. One company was able to identify the resistant allele to IPN. 2 generations, they were able to exclusively produce eggs with only the IPN resistant gene, and the mortality dropped to 0. So in 2 generations, the mortality due to IPN went from 40% to 2%. In the last 10 months, there's a new technique that has been developed called CRISPR. And what this does is allows a laboratory or a technician to slice the chromosome in specific spots. So if a gene is located for white spot resistance or MS resistance, the company can use CRISPR to take out the bad allele and put in the new allele. And as a result, in just a couple generations, you can go from high susceptibility to high resistance. It is a very powerful technique that has been demonstrated in laboratory animals. The advantage of CRISPR versus some of the other techniques that have been used in the past is with CRISPR, you're introducing genes or alleles from the same species. So it appears that the use of CRISPR will not cause an animal to be labeled as GMO. Also, with the sequencing of the shrimp genome, it is going to be possible to relate the genetics work in other species to the shrimp, and before long we'll be able to control some of the gene functions and learn how to turn on a certain set of genes, and by turning on genes we will be able to produce animals that are more resistant to pathogens. Muchas gracias.
Gracias de nuevo, Richard. Bueno, con esto cerramos la jornada de la mañana. El almuerzo ya está servido. Regresamos 1:30 tal como está en el programa del evento. Provecho.